Welcome, student, to the Histology lab of the digestive system. In this first part, we are going to talk about the histology of the alimentary tract, including the tongue, esophagus, stomach, the small and the large intestine. So we start with the histology of the tongue. First, this image shows a section through the tongue, general view of the tongue, the histology of the tongue, showing several features. We have epithelium, we have glands, here glands and here glands, and we have muscles. And of course, one must not forget that in the tongue, we have papillae. So we are going to look at these structures and these features in more detail. In these images here, the first one here, we have the epithelium of the tongue. The tongue is covered by stratified squamous epithelium. Most of the tongue is covered by stratified squamous non-keratinized epithelium. As you can see here, this is the epithelium, and it is stratified, and the topmost layer is squamous, and it is not covered by keratin, so it's stratified squamous non-keratinized epithelium. Also, as you know, the tongue has muscles, and these muscles are skeletal muscles. So how do skeletal muscles appear? In a longitudinal section, like these, we see that first of all, we have nuclei at the periphery, as can be seen here. And the most important feature is that in the longitudinal section, the skeletal muscle fiber appears to be striated with alternating light and dark areas, as can be seen here in this image. What about in a cross-section? These are cross-sections of skeletal muscles of the tongue. In a cross-section, since the nuclei at the periphery, so we find the nucleus at the peripheral part of the section. Okay, as can be seen clearly in these images. So here we have both longitudinal and cross sections of the skeletal muscles of the tongue. In this image here, we can see glands that are found in the tongue. We have both mucus and serous glands, and the mucus glands are characterized by having the cell with no color. Cells are devoid of color, whereas the serous glands, these are the serous glands, they have color. The characteristic feature of the epithelium of the tongue is that it has special structures called papillae. And we have several types of papillae. This is the first type, the filiform papillae. Filiform papillae are sharp conical structures covered by stratified squamous keratinized epithelium. So this is the epithelium, and this is the keratin. As we have said, most of the tongue is covered by stratified squamous non-keratinized epithelium. So why are the filiform papillae covered by keratinized epithelium? Because these papillae are used to manipulate food. They are used to move food particles during swallowing and while chewing inside the mouth. The fungiform papilla, as can be seen here, is another type of papilla found in the tongue. Here, these papilla serve for taste sensation, and therefore they have taste buds. The taste buds in the fungiform papilla are found on the apical surface of the papilla. Here, this is a taste bud. This is a taste bud, and here is another one. So this is the fungiform papilla, of course. This is the epithelium, stratified squamous non-keratinized epithelium, and this is here we have connective tissue, as usual. The last type of papillae is the circumvallate papillae. These are the least numerous, but they are the most important. These are large and dome-shaped, as can be seen from the image. They are dome-shaped. Now, these are the main site of taste sensation. Although they are the least in number, they have the most taste buds in them. And the taste buds are located on the periphery of the papilla, on the peripheral side of the papilla, and not on the apical. In the fungiform, it was found on the apical surface, whereas for the circumvallate, the taste buds are found on the periphery of these papilla. Also, we have serous glands found nearby these serous glands, and they empty their secretions into the furrows surrounding these papillae. So as you can see here, we have some kind of depression or a furrow surrounding the papillae, and within it, the serous glands produce their secretions. So all these are glands. These are all glands, and the secretions are produced in this furrow. Of course, these wash out the molecules so that the taste bud can perform its function again and again. This is, of course, the epithelium, and this is the connective tissue. Here is a magnified image of the previous slide showing the taste buds. So taste buds are oval structures, oval in shape, and they are formed of several types of cells. The gustatory cells, the supporting cells, and the basal stem cells. Now, it's not easy to differentiate between the different types of cells in light microscope. However, if we look at these nuclei, which are located at the bottom of the taste buds, these are the nuclei of the stem basal cells. But it's not important for the practical to identify the different types of cells in a taste bud. What's important is that the taste bud has a round opening at its apex. Here and here. These round openings are called the taste pore. Through it pass what we call taste hairs. As you can see, only can be seen here. These are a tuft of taste hairs. The taste hairs are actually microvilli of the gustatory cells, and these are the structures that are responsible for detection of the various chemicals found in this region to produce taste sensation. So, taste buds are oval structures with different type of cells. They have a round taste pore through which taste hairs will pass. Now let's look at the histology of the esophagus. Here we can see the typical structure of the wall of the organs of the alimentary tract. And these organs are lined by a wall formed of 4 layers. And these are, starting from the layer closest to the lumen, we have mucosa, This part here, this is the mucosa, and then we have the submucosa, then the muscularis externa, and finally an outer layer of either an adventitia or serosa. Now the mucosa is formed of epithelium, lamina propria, and muscularis mucosae. The epithelium in the esophagus here is stratified squamous, non-keratinized epithelium. Under it we have the lamina propria, which is a connective tissue layer, and the last part of the mucosa is the muscularis mucosae. It is a thin layer of smooth muscle cells that separates the mucosa from the submucosa. The submucosa is a connective tissue layer, another connective tissue layer. And then we have the muscularis externa. The muscularis externa and the different organs of the alimentary canal differ in the number of layers within the muscularis and the type of muscle. In the esophagus, we have an inner circular and an outer longitudinal muscle layer. And also we must not forget the muscularis externa contains the myenteric plexus of nerves, which is part of the enteric nerve system. And the submucosa contains the submucosal plexus of nerves. The outermost layer could be either an adventitia or a serosa. If the organ is surrounded by peritoneum, the outer layer is serosa, formed of connective tissue and epithelium. And if the organ is not covered by peritoneum, then the outermost layer is connective tissue adventitia only. And of course, the esophagus is a thoracic organ, therefore it is not covered by peritoneum, so the outermost layer is connective tissue adventitia. This is another view of the esophagus. Here we have the mucosa formed of epithelium, lamina propria, and here we have the muscularis mucosae. This is the submucosa. As you can see, it's a connective tissue layer, and I can see several glands in them. These are all glands in the esophagus. And after the submucosa, I have the muscularis externa. The muscularis externa of the esophagus is formed of an inner circular layer and an outer longitudinal layer. This is the outer longitudinal layer. Finally, the outermost layer of the esophagus is connective tissue adventitia. Now, this image is actually of the upper part of the esophagus. In this part of the esophagus, the muscularis externa is formed of skeletal muscles. And how do I recognize that in the image of the slide? Now, this is a cross-section through the esophagus. When I take a cross-section through the esophagus, this means I'm taking the cross-section through the esophagus, and I have the circular muscle. This means that I am taking the longitudinal section through the muscle fibers, because the circular run along the diameter of the esophagus. So actually, I'm taking the longitudinal section through the circular muscle layer. So how do they appear? Long cells with nuclei at the periphery, and I have cross striation. What about the longitudinal layer? When I take a cross section through the esophagus, I am actually taking cross section through the longitudinal layer, because the longitudinal layer is running along the length of the esophagus. So when I cut the esophagus transversely, I am cutting the longitudinal muscle layer also transversely. So how will I see them? I will see them as circles. And here the nuclei will appear on the periphery. Okay, in the middle of the cytoplasm, we will not see any nuclei. So this is how I know this is a skeletal muscle. And if I see skeletal muscle, this means that this is the upper part. Here is a cross-section through the lower part of the esophagus. In this part, the muscularis externa is formed of smooth muscle. So when I take a cross-section through this part of the lower part of the esophagus, this means that I am taking a longitudinal section of the circular muscle layer. So I will not see any striation. When I examine the circular layer, I will not see any striation here because it is smooth. But in the longitudinal muscle layer, I will see again the circles. But here, the nucleus will be in the center. The nucleus will be in the center because these are smooth muscle cells and the nucleus is in the center of these cells. So this is how I differentiate between the upper and lower parts of the esophagus by looking at the muscularis externa. If I see the externa formed of skeletal muscles only, this means that I am in the upper part. And if I see the muscularis externa formed of smooth muscles only, this means that I am in the lower part. Remember that in the middle part of the esophagus, I have a mixture of both skeletal and smooth muscle cells. The gastroesophageal junction. This is the region of the junction. This is the esophagus on the right side, and here we have the cardia of the stomach on the left side. Notice how we have a change in the epithelium, a change in the lining epithelium, although the other layers are almost continuous with each other, but we have a change in the epithelium. And if you look at the magnified view, we see that we have a sudden change. Suddenly, at this region, the stratified squamous epithelium of the esophagus has changed into the simple columnar epithelium. All these are glands. All these are glands of the stomach, and the change is sudden. So the stratified squamous epithelium of the esophagus has suddenly stopped, and the simple columnar epithelium of the stomach has begun. The histology of the stomach. Now, as in the rest of the alimentary tract, the wall of the stomach is formed of mucosa, submucosa, muscularis, and the outermost layer in the stomach is a serosa. Since the stomach is an intraperitoneal organ, the outermost layer is formed of serosa, which is connective tissue and epithelium. The mucosa again is formed of epithelium. This is the epithelium of the stomach, and we have connective tissue lamina propria. Of course, although we are seeing the lamina propria under the epithelium, but it actually also between the epithelial glands of the stomach. And then we have the muscularis mucosae, this thin layer of smooth muscle cells that separates the mucosa from the submucosa. Submucosa, of course, is connective tissue layer. The muscularis externa of the stomach is formed of three layers. Now, in light microscope, in most slides, it's not easy to differentiate between these three layers. So we can only see that this is the muscularis externa. Serosa is formed of connective tissue plus epithelium. And epithelium, of course, is simple squamous called mesothelium. These are two images of the wall of the stomach also. In this one on the left, this is the mucosa formed of epithelium. This is the epithelium, and everything that is whitish below and between the epithelial glands, this is the lamina propria, and this thin red line, this is the muscularis mucosae. This layer here is the submucosa. This is the submucosa, connective tissue, and here we have the muscularis, and again you can see the serosa formed of connective tissue plus epithelium. Here is another image of the wall of the stomach. This is the epithelium. Between the epithelium and under it we have the lamina propria connective tissue. This is the muscularis mucosae. Here we have the submucosa, the muscularis externa, although not very clear, but the serosa is supposed to be the outermost layer. Now we must remember that in an empty stomach, the wall is thrown into folds called rugae, and this image is of a ruga, and the rugae are formed or are folds of submucosa and mucosa. So, as you can see, we have folding here of the mucosa with the submucosa. Now, you can clearly see here we have the epithelium and the lamina propria, and this is the muscularis mucosae. Now, there are many cells in the epithelium of the stomach, and we have many types of cells in the gastric glands. However, in the lab, we are only going to talk about few of them. The easiest cell of the gastric epithelium to identify under the light microscope is parietal cells. Parietal cells, because they are large and because they are acidophilic, they are easy to identify. So all these acidophilic structures, all these acidophilic cells, these are the parietal cells. These are parietal cells, large acidophilic cells. You must remember that they are acidophilic because they contain several mitochondria. And by the way, the basophilic cells, like this one here and these ones here, these are chief cells. These are chief cells. Chief cells are basophilic. Another image here. Again, the red cells, the acidophilic cells, these are parietal cells. As you can see, they are easily identified and they are numerous. The cells with the parietal that appear vacant or that are white in color, these are the mucous neck cells. All these are the mucous neck cells. Okay. Now, we must also remember that since we are seeing a lot of parietal cells in this image, this means that most probably this section is from the fundic glands, from the fundus or the body of the stomach, because as we have said, that in these two regions we have mainly parietal and chief cells. Histology of the small intestine. We start with the duodenum. Again, we have mucosa, submucosa, muscularis externa, and the outermost layer is either serosa or adventitia, depending on whether that part of the duodenum is intraperitoneal or not. Now, the mucosa here, we must remember that in the small intestine we have folds of mucosa and submucosa, the plica circularis, and the mucosa is thrown in, has villi, but in the duodenum these two features are not very prominent. The characteristic feature that make us easily identify the duodenum is that in the submucosa we have several glands. All these are glands, mucus secreting Brunner glands, okay, found in the submucosa of the duodenum. For the muscularis externa, we have an inner circular and an outer longitudinal muscle layer. Here is another image of the duodenum. Here we have the mucosa, and this is the submucosa. Notice the several glands that are found here. All these are submucosal Brunner glands. The jejunum, here in the jejunum, we can easily identify the plicae circularis, and the villi are most prominent. Remember that most of absorption takes place here in the jejunum. So here we have a fold of mucosa and submucosa. So this is a plica, and these finger-like projections, these are the villi. As you can see, they are easily identified and they are prominent in the jejunum. Between the villi, we have the intestinal glands. So these structures reaching deep down, almost to the submucosa, these are the intestinal glands. Of course, the muscularis externa is inner circular and outer longitudinal, just like the duodenum. For magnified view, here we have a villus. This is a villus. Of course, it's formed of epithelium, and the epithelium is simple columnar epithelium. And this is the core of the villus, which is the lamina propria with all of its content. Between the villi, reaching deep down, we have the intestinal glands. All these are the intestinal glands with its various cells. Again, this is a further magnification of a villus. Here we can easily see the simple columnar epithelium. Notice the oval-shaped nuclei of the columnar cells. Also, what we note here, we note these empty-looking cells. Empty-looking cells. This one here, this one here, here. These empty-looking cells are goblet cells, mucus-secreting goblet cells. And as we have said several times, mucus disappears during preparation, and therefore these cells appear empty. Also, what do we note? The surface of the epithelium is covered by this thin red-looking line. This is the brush border of the intestinal epithelium. Brush border. It appears like a brush, the hairs of a brush. And this brush border is formed by the numerous microvilli that are found on the surface of the small intestinal epithelium. The last part of the small intestine is the ileum. The characteristic feature of the ileum is that we have large lymphatic nodules in the submucosa, reaching deep down into the submucosa of the ileum. These are Peyer's patches. Okay, so this is the characteristic feature of the ileum. This is another image. Here we have the mucosa, here we have submucosa and the muscularis externa, and the outermost layer is the serosa. Again, we can see in the submucosa we have the numerous lymphatic nodules. So, to differentiate between the three parts of the small intestine, for the duodenum we have the numerous glands in the submucosa. All these are glands. In the jejunum, we have several plicae and prominent villi. We can easily identify the plicae. These are all plicae. And as you can see, these are all villi, prominent villi. For the ileum, we have the submucosal lymphatic nodules, the Peyer's patches. This is the characteristic feature of the ileum. Finally, the histology of the large intestine, specifically the colon. Again, we have the mucosa, submucosa, and the muscularis externa. The outermost layer could be either a serosa or an adventitia, depending on whether the part of the colon is intraperitoneal or not. Now, here we can see the characteristic feature of the muscularis externa of the colon. First of all, here we have two layers, an inner circular and an outer longitudinal. However, the outer longitudinal layer does not wrap around the entire wall of the colon, but it is bundled together in three bundles called tenuculi. So here we can see a bundle of the outer longitudinal layer of the colon. Here is a cross section through the colon. We can see this is the mucosa, this is the submucosa, and here we have the muscularis externa. We can see here we have a bundle of tenuculi, and this is another bundle, and here is another bundle. So the tenuculi, the longitudinal muscle layer, have arranged themselves into three longitudinal bundles of muscles. This is a characteristic feature of the colon. Also, what do we see here? We see a lot of lymphatic nodules. Lymphatic nodules found in the submucosa, reaching down deep into the submucosa of the colon. Here is a magnified view of the mucosa. Of course, we do not see any villi here. We only see the glands. All these are the intestinal glands, the glands of the large intestine. Characteristic feature of these glands is that we have numerous goblet cells. You can see that most of the cells are empty or appear white because these are all goblet cells. So most of the cells here are actually goblet cells. Here we can see the muscularis mucosae very clearly. This is the submucosa, and here we have the muscularis externa. And by this we end the lab about the alimentary canal. See you next time. Thank you.